Hello guys, I am Shubham from ShubhVed tutorial. This video is about tetrad analysis. So let's get started with the video. So basically, what is tetrad? That we'll see first of all. So this is seen basically in fungi, lower eukaryotes, usually fungi. Okay, lower eukaryotes. Okay, usually fungi. Right. So we know that fungi are haploid. Means means they have one set of chromosomes. Okay, as we as a higher eukaryotes are diploid. So fungi are haploid. They follow asexual as well as sexual reproduction. So we here are concerned with sexual reproduction. So we will see how this occurs. Okay, so basically, what is haploid condition? See, I will draw here one cell in which there is a chromosome, right? Okay, one more cell I will draw here. So these are the two haploid cells. Okay, then they will get fused. They will get fused to form a zygote. Okay, this is a zygote having 2n, right? See, these are the two chromosomes fused here. Okay, so this is haploid, and this fusion is known as diploid zygote. Okay, so now what happens? This will undergo replication. This will undergo replication. We know that what is replication? I mean, there will be duplication of chromosomes. So after that, they will form a structure like this. Okay, this is one chromosome after duplication, and another is with, right. So these are the two chromosomes replicated. Then this will undergo meiosis. Okay, then it will undergo meiosis. Meiosis 1. Meiosis 1. After meiosis, what will happen? I will just draw here. Then there will be two separate chromosomes will get segregated, right? After the meiosis 1. So like this, the structure will be formed. Suppose this is one cell and this is another cell, right? And the chromosome getting separated here. Okay, so this is arrangement after meiosis 1. Now this will undergo meiosis 2. Again, meiosis 2 will undergo meiosis 2. After meiosis 2, what happens? The sister chromatids will get separate, right? I will draw your big circle as 1, 2. 3, 4, right? Okay, so after this will have the one chromosome red. Okay, right? So this is the arrangement of 4. Okay, after meiosis 2. So this arrangement is known as tetrad. Okay, so this is tetrad, right? So after this, the some species undergo mitosis. Okay, undergo mitosis. After this, I will just make a space for easy explanation. Okay, so after here, mitosis happens. What happens? Mitosis, and mitosis gives rise to 8 cells. Then 4 and 4. We know the mitosis. This is one. I will draw small. 3, 4, 5, 6, 7, 8. Okay, and enclosed in a sac. This is known as sac. So now 4 are red. After mitosis, 4 are red, right? Okay, so this is this arrangement after mitosis. There are 8 spores, right? These are known as spores. So this 8 arrangement is known as octad, right? So this is a overall pathway of how the tetrad, what is tetrad and what is octad, right? So this is basic sexual reproduction of fungi. Okay, now I will just tell you that this inside this the outer sac in which this all gametes are enclosed. Okay, this sac is known as ascus, right? This sac is known as ascus. Okay, so this is known as ascus, the outer sac. Okay, and the initial, this all the things are known as spores. Okay, we know that this if this is broken, the spores are released. Okay, so these are spores. So spores are enclosed within a sac, right? So this okay, so all this are known as spores. So the fungi that forms this are basically ascomycetes. Okay, ascomycetes, ascomycetes, right? So this is a tetrad basically. Now, depending on the arrangement of this tetrad spores, they are classified as ordered and unordered tetrad. Okay, ordered and unordered. So what is ordered? As we saw here, as we saw here, as we saw here, this arrangement, okay, this arrangement is known as ordered. Okay, ordered means what? Linear arrangement of spores. Linear arrangement of spores. Okay, in order there is a linear arrangement, and in unordered tetrad there is a random mixture. Okay, I will show you here. Suppose this is a, okay, then I will draw here. Okay, in this arrangement is anyhow. Okay, so this we called as random. So this is known as unordered tetrad or octad. Unordered. Unordered is what? Random arrangement of spores. Okay, so there is a random arrangement, right? And this type is seen in Saccharomyces cerevisiae. Saccharomyces cerevisiae. Okay, and the other type is seen in Neurospora. Ordered tetrad is seen in Neurospora crassa. Neurospora crassa. Got it? So these are the two examples. Okay, ordered is linear arrangement, unordered is random arrangement. Random arrangement is seen in Saccharomyces, and ordered arrangement is seen in Neurospora. So basically these are used, okay, for study of genetic map to measure distance between centrosome and centromere and a gene. Okay, so basically this is a chromosome. As we all know, this is a chromosome here, and this is a centromere. Suppose this is a here is a gene A. So this distance we can find using this analysis. Tetrads. Okay, so this is used for that mapping, and mostly Neurospora crassa is used because there is a linear arrangement. Okay, suppose these are the two AA genes. We know that I will write here that see, so this is a A gene here, red, and this is small a. Okay, so after separation we know that, and this is linear. This is A, this is A, this is small a, this is small a, right? And all these are small a's in octad, and here capital A, right? So mostly this is used. Neurospora experiment are done, okay, for analysis. So basically, this was about basic idea about tetrad, what is ordered tetrad and what is unordered tetrad. In the next video, we will see about detail on ordered tetrad analysis. Okay, so that's it for this video. I hope this video helps you. If this video helps you, do let us know in the comment section. Thank you guys for watching this video. If you like it, please share it. Don't forget to subscribe and press the bell icon to get notified whenever the video is out. Also follow us on all our social media pages, and you can join us on Telegram and stay connected for the latest updates. All links mentioned in the description section.